This experiment session. So today we are going to have two experiments simultaneously. So one is your SDS Page and other experiment is two-dimensional polyacrylamide gel electrophoresis. So before going on to the workflow chart of these two particular experiments, I think you should know what are the basic differences between these two. So once you know the differences, then only you can think of how to use this. For example, SDS Page. This is very user friendly if you compare with the 2D page. Then SDS Page is very cheap as compared to 2D page. SDS Page you need to require one instrument, whereas in case of 2D page, since this is two-dimensional, you need to have two different instruments. In case of SDS Page, the basic principle is molecular weight. So based on molecular weight, you are going to separate the protein. In case of 2D page, so you are going to use two different mode. One is isoelectric focusing, which is based on the isoelectric point of the protein, and other is the molecular weight, which is basically the SDS Page, which you are going to do. In case of one-dimensional SDS Page, so for protein profiling, SDS Page is the best tool. For protein profiling or protein pattern profiling, SDS Page is the best tool. For the identification of protein, 2D is the best tool. In case of SDS Page, it's difficult to identify the protein. You can analyze the pattern, but you cannot identify, it's very difficult. But in case of 2D page, it's easy to identify or establish the identity of the protein. In case of SDS Page, very few proteins can be accommodated on the gel, but 2D page gives you power to accommodate more proteins on the gel. 2D page has downstream applications. SDS Page has very limited applications. SDS Page can be used for Western blotting also, right? So once you run the SDS Page, then you can screen with primary and secondary antibody to execute the accumulation pattern of protein. In case of 2D, it's difficult because this is protein spots, so it's difficult to hybridize the antibody. SDS Page is cheaper one. 2D Page is costly. In case of 2D page, you need to have IPG strips. In case of SDS Page, there is no requirement like this. In case of SDS Page, so this blue staining that you can do, normal staining what we are doing in lab, but in case of 2D page, you can go for blue staining, it's easy, but then high resolution figure will only come when you go for silver nitrate staining or silver staining. Right? So these are some of the differences between these two. Regarding uses, SDS Page is routinely used for protein profiling. 2D is routinely used for establishing the identity of the protein. These two things you should keep in your mind. Now let's take these two experiments one by one. So first is SDS Page. As I told, in case of SDS Page, the basic principle is molecular weight. As the name stands, this SDS has come from sodium dodecyl sulfate, which is basically a anionic detergent, right? So when you add SDS, so this SDS has a very peculiar characteristic that it will, suppose this is your functional protein, when you add the SDS, so it will linearize your protein with a uniform charge, or charge is unified in case of SDS Page when you treat your structured or functional protein with anionic detergent. So this anionic treatment is done with a beta mercaptoethanol, so which will break the disulfide bond, so that your protein should be in the linear form in having a uniform charge, and when you run this on the gel, then the separation should be based on molecular weight. All right. So there is a term which is very popularly used in case of SDS Page, that is called as band sharpening effect, right? So what happens is, many times you have seen when you run your sample, so band will come, but then it will be blurred one, it will be not, the clarity will be not there, right? So a term has been coined, that is band sharpening effect. So what happens is, when you make a gel, Suppose if this is a gel, so as you all know, SDS Page is also called as two gel system. It is also called as two gel system. So this is your first gel which is stacking. This is your another gel which is called as resolving. Stacking means when you load the protein, your protein is going to be stacked in the stacking gel. Resolving means once protein is stacked, it will move to the resolving and it will separate in the resolving gel. So these two, since we are using two gel, so this is also called as two gel system. It is also called as discontinuous buffer system, because you are using two different type of buffers at two different chambers. Discontinuous, there is no continuity in the buffer which you have accumulated. This is also called as 3 pH system. Why 3 pH? Because in case of stacking, the pH which you are going to use is 6.8. In case of resolving, the pH which you are going to use is 8.8, and the running buffer, the glycinate buffer which you are going to use, has a pH of 7.3. And let me tell you, SDS Page is all about pH. This is very important information. Most of the time we take precaution of everything except pH, and your result 99.9% depends on the pH of the solution which you have made. Even if you fluctuate this with 2 digits, so it will have an effect which is clearly visible on the band resolve. So band will be not clear, or there will be some other problem. So pH should be accurate as written here. So because of this, it is also called as 3 pH system. It is also called as Lamley system, because Lamley is the name of the scientist who discovered the buffer of this, and he showed the running pattern of SDS Page. Lamley's, Lamellis, Lamley's buffer system. So band sharpening is that when you load, so this is your stacking gel, this is your resolving, and these are the wells. There is a thumb rule that the well bottom and the transition of stacking and resolving, at least 1.5 centimeter space should be there. At least that much distance path length should be there, so that your protein should travel at least 1.5 centimeters. So what happens is, when you load your sample into this well, your sample will just accumulate here, and then when you provide the current, right? So since this stacking gel is at very low concentration, most probably we are using 4% polyacrylamide gel here for making the stacking gel. So your protein will enter inside the stacking gel, and in your buffer you are using an ion which is called as glycinate and chloride ion, right? So what happens is, once your protein enter inside, so these ion will charge, and because of a pH of 6.8, so this glycinate ion, it will start giving a dragging force, right? Because the electrophoretic mobility changes at 6.8 pH. It will be almost neutralized, and once it neutralizes, so it will show its movement in a dragging way. So one ion starts showing the dragging, whereas at the same time chloride ion, it will start showing high electrophoretic mobility, right? So one is running at fast speed, other is stopping it, and because of this your protein will sandwich in between this, because there will be a ripple. One ion is running, giving a thrust, other is stopping it. So this ripple stop this or sandwich the proteins, and with every ripple this protein will start accumulating, squeezing, and there will be a slow movement also, and once it reaches at this phase, by the time your protein will sandwich and it will form a uniform pleated structure. Once it enter inside this, now here the concentration is almost 9% to 12%, and in some lab people they are using 14% also in the resolving gel. So 9% or 14% means the pore size is very small one. So once this pleated structure of protein reaches here and it tries to enter inside, so it requires a lot of force, and once it enter inside, suddenly pH will change to, now reverse happens. right? Now reverse happens, so now both ion, the rippling effect will be neutralized, and now from here onwards the movement of the protein will be purely based on the electric field. And since the gel which you have used has very high concentration of polyacrylamide, so the pore size is small, so now sieving effect will start. Sieving means that pore will filter the protein, and based on its strength and based on the molecular weight it will start moving. It is because of this reason that you are getting a protein band at different locations, right? Now few precautionary measures which you should know before executing the SDS Page. First important thing, as I told, is pH. You cannot manipulate with the pH. pH should be strict, what has been written over here. So it will have direct effect on your result. So pH, that you should be extra cautious. Second is the gel, the resolving and the stacking gel. So the chemical which you are going to use, that should be high quality biochemical grade chemicals, right? Because lot of contaminant based chemicals are available. So if you compromise with the chemicals, most of the lab I have seen, so if you use below standard chemicals, so your run and protocol is very perfect, but they complain that's a result, that clarity is not there, right? You have to use standard chemicals. Third is gel preparation. So there is a term which is very popularly used when you talk of polyacrylamide gel preparation, that is called as degassing, right? Now please ask yourself how many times you have done degassing of the chemical before going for SDS Page. I don't think so any one of you do. So degassing is a very critical step where you are going to remove the oxygen molecule from the solution of acrylamide and gel, because if oxygen is there, it will hinder the polymerization of your gel. So when you degas it, when you remove the oxygen, then your gel is perfect, and when it polymerizes, there will be uniform polymerization from top to bottom. So many times when you see with your naked eyes, so top very perfect polymerization will be there, but between somewhere bottom corner it will be in a pulpated state. So when you load your sample, you can see your protein, first it will be in a length, and then there will be bulge, and then it will be a big eye like bulge. This only happens because uneven polymerization. So that you have to take utmost care. Polymerization should be uniform and perfect. Second, when you are loading, suppose this is a gel. Now I have to load. So always the filling of this should be from one corner. From one corner you have to drain. If you put your liquid, that acrylamide, from the middle, so it will take bubbles also, and then it's difficult to remove the bubbles. And if bubble is there, bubble is like a pore, and it will just divert the electric field which is going to create in your gel, right? Third thing is, once you have poured this and now you have kept it for polymerization approx for 30 minutes, so then this particular washing, this transition should be uniform one. So people, they are adding isopropanol, few people they are adding water itself. Even if you add water, that will keep it uniform. If it is not uniform, 100% you will not get the results, right? So you can add water, you can add any organic solvent. So organic solvent, you will make a uniform layer, and once it polymerizes, so the layer will be uniform. Fourth point is, once you are draining the liquid, then you have to keep it upside down so that whatever organic solvent or water is there, that should drain out. It's not that I have just once upside it down, then I load the stacking. It means that stacking and resolving transition will be not perfect. If it is not perfect, what happens is, when your sample reaches here, it will drain this side. So it will give a look of chapati. It will drain both sides. So that we have to take precaution, right? And last point is, once your gel is, once this is polymerized, when you load this stacking, This is our comp. This distance you have to maintain. If distance is not maintained, minimum 1.5 centimeters, so what happens is that travel area will be less, so your protein will be not stacked. If it is not stacked, so you will get a smearing. Many times if you recall your result, you have get a smearing in the lane. This is because of manipulation in this particular area, right? So with this brief, let me take you to the workflow chart of SDS Page. So first thing is requirements. What are the things which you require? So as I told, the first thing which you required is instrument. SDS Page unit. Basically we have three different type of SDS Page unit. The first is mini gel system. Mini gel system. This is a 7 centimeter mini gel system. This will cost you around 20 to 25,000 rupees. This is most popularly and routinely used in the lab. So cost is 20 to 25,000 rupees. It takes 2.5 hours to complete the run. 2.5 hours to complete the run. Then next is MIDI system. MIDI gel system. So this will cost you around 75 to 1 lakh rupees. It takes 4 to 5 hours to run. 4 to 5 hours to run. And it is 12 centimeter, or maybe you can use 16 or 17 centimeter also. Then last is large gel system, right? So here you can use 18 or 24 centimeter, and it takes almost overnight. It takes almost overnight. You need to have a dedicated coolant with this. So many times you have to, many tubes will be there in the SDS Page unit. That is basically a cooling chambers. So water will go on circulating and then it will keep it cool, right? And this will cost you around 2 lakhs rupees, 2.5 lakhs rupees. This mini gel is used routinely. MIDI gel is used when you need to have a resolution of the protein which you are going to separate. And this large gel is used when your objective is identification, characterization, establishing the identity. So in that case, people, they prefer to use large gel system. And as the name suggests, as you increase this side, the ingredients or the requirement will also increase. Second is sample preparation. So when you talk of SDS Page, so there is a different way of sample preparation. The most common way is that you extract your sample in a 100 millimolar Tris-Cl buffer, and pH should be 7.3. This is thumb rule. If you don't know, if you don't have information about how to extract, so you can use this extraction buffer, 100 millimolar Tris-Cl, pH 7.3. Extract any tissue except the seed, right? You extract it, centrifuge, take the supernatant and run it, you will get very beautiful pan. But if you are doing precise work, then you have to be very particular. Like different kits are available. Like we have a focus proteome kit. Focus proteome kit. This is proprietary of G Bioscience. Beauty is you can purify as well as you can extract the protein. You can purify also, you can remove all the contaminants. And this is very easy, within 10 minutes you can do. This is column based method. We have a 2D extract kit. This is also a proprietary of G Bioscience. A very good kit, routinely you can use for the extraction of your protein from plant tissue. 2D extract kit. Okay. We have quantitation kit. This is proprietary of Amar Sham. So here you can extract as well as you can quantitate, quantify the protein. Amar Sham. Right? And like this, n number of companies are there with their own product. You can use any of those kits to isolate the protein, right? And if you have a specific requirement, for example, those who are physiologists, they might be knowing that when you are dealing with leaves, Rubisco is one of the major problem because it is most abundant protein. So you need to have a kit which can remove the Rubisco, because if Rubisco will be there, it will interfere with the other proteins. So you have to remove the Rubisco. So we have a kit for this, which is called as Rubisco exclusion kit. So just pass your sample from that, Rubisco will be out, rest other protein will be in the fractions. So once you isolate the protein, then comes quality analysis. So without quality analysis, it's something like dubious to use this for your experiment. So quality, whatever instrument I have listed, you can use any of that for the quality analysis. You can use bioanalyzer, you can use nanodrop, you can use qubit. So all these instruments are dedicated to quantify as well as tell you the quality of the protein which you have isolated. Once you are assured that the concentration and quality of my protein is okay, then comes running part, how to extract and run the sample, right? So this is your plant. So you have isolated your extract. So here one precaution which you can write in footnote. So when you crush your sample in liquid nitrogen, you have to take precaution that you have to give thrust, less of liquid nitrogen, more of thrust, and this should be done till you get a powdery talc-like structure. Many times if this is partial or if some particles are still left over, it means that much representation is out from your extract. So thumb rule is less of liquid nitrogen, more of pressure, and you have to repeat till you get a powder-like, fine powder-like structure in your pistol mortar. Then comes centrifugation. In case of protein, this is a difference between proteomics and molecular biology what you are doing. Here the speed should be 15,000 to 18,000 RPM and minimum time should be half an hour. Minimum time is half hour in order to have from a small molecular weight to high molecular weight protein. So many times it happens that we'll go for 12,000 RPM for 10 minutes like what we are doing in case of molecular biology. Let me tell you 50% of the protein will be in tissue itself. Only you will get 50% protein out of your extract, right? So in case of proteomics, duration is very important, it should be high, more. So once you extract this, then you have to go for quality. Once quality is okay, then you have to go for quantity. So there are n number of methods to quantitate the protein. If I go back, Lowry's method was one of the most popular method. Then now Bradford method is there, which will tell you all the soluble protein which is present in your sample. If you don't want to use Bradford, then you can use qubit. If you don't want to use qubit, you can use nanodrop. If you don't want to use nanodrop, you can go for bioanalyzer. No need to go further because this is the costliest one which you can use for the quantitation of the protein. Once you quantify this, now you are ready with your sample. The first step is that you have to take your protein of equal quantity. Suppose if I have a control and treated sample, so you have to take protein of equal quantity, right? Then add a loading dye into this. Add a loading dye into this. Then you have to denaturate it. Denaturation of protein. That we are doing by heating it at 95 degrees Celsius for 5 minutes. This is very important. 95 degrees Celsius for 5 minutes. So you can use any of the dry blot. So there you can, dry heater is there, there you can keep 95 degrees Celsius for 5 minutes. Then take a pen drop and immediately keep it in ice. Take and immediately immediately keep it in ice. Now this is ready to be loaded. Now this is ready to be loaded. Now this is the gel. Many times it happens that when you take the comb out, right, your gel will start displacing, right, or many remnants will be there. A person is not supposed to load sample until unless you clear the remnants and you make it straight. Easily you can do. The comb you can turn it. If you have the comb like this, thick, you turn it and from back you can make it straight. All your walls should be straight when you are running SDS Page. First thing. Second thing, if some remnant is there, many times when you remove a comb, some remnant will be there. So in your lab, Whatman paper is there, right? You take or make a thin strip of Whatman paper like this. Cut using scissor, thin strip of Whatman. Then put Whatman inside and in one go take it out. All these remnants will stuck onto the functional group of Whatman and it will, in one go it will come out. I mean to say, well should be neat, clean and straight. Then only you will get the best quality results. So once your well is clean, then you load the sample, feed the instrument into the running tray and then run it. Right? So once run is complete, now you have to tell me how you are going to know whether the run has been completed or not. So there is a tracking dye in this, bromophenol blue tracking dye. So based on the movement of the dye, once dye reaches somewhere at the bottom, you have to stop your run. So in case of mini, it will take 2.5 hours, right? So once your run is complete, from one corner using some thin strip or maybe sometimes spatula also, from one corner you just give a jerk, take a water in a tray. This is your tray, fill it with water, take this plate, put it angular like this, then put a spatula at one corner and just give a jerk. Once or twice you give a jerk, that ripple will be absorbed by the water and this plate will come out. Because this is the most tedious step where most of the plate broke out when you put pressure to remove that upper plate. So take a water, put plate inside, give a jerk here. Once or twice when you give jerk, upper plate immediately will come out and water will absorb the ripples. Once it is out, then it is ready to be staining. You can use, and nowadays ready-made blue safe stainings are available. You just put it in the staining, keep it for 2 hours and then you can use methanol and then water, glacial acetic acid for destaining. Once you destain, you will get a band or a result. Okay? But the informative part is you should know to troubleshoot the result, right? Let's have few situations that how to troubleshoot the result which you are going to get. Now this is my SDS Page gel. These are the lens. First is basically for the marker, right? Now in this lens I am getting protein like this. Now you have to tell me what may be the condition, what happens to this gel. That at initial we got certain plumbing, but then rest all are little bit faint proteins are there. Yes, the reason is the protein which you have used is degraded, right? Or you will get protein like this, partially degraded, or you will get protein what may be the reason? Smiley face. What happens? Huh? No. Exactly. The voltage which was expected, you have used more than what was expected. And the outcome of such type of run is, many times you have seen when you take the plate out, it will be broken and you will be surprised how it happens. It happens because you have used high voltage, and let me tell you, for every 1 degree, 1 degree Celsius increase in temperature, so when you use a stretch of 5 volt increase, for every that 5 volt will increase 1 degree Celsius temperature inside the gel. It is because of this reason that we need to have a coolant. And when a voltage reaches a certain parameter, so this plate automatically will broke out, shattered. Many times it happens that you will get a protein like this. Now upstairs very beautiful band pattern is there, but there is a dragging and then nothing is at the bottom. What may be the reason?
Loading error.
No, if loading error would have been then here, you would have not got the protein bands. So there is two reasons. One is that maybe at this step itself we stopped running. Second reason is the gel which you have prepared, most probably this lower part is not casted perfectly. Lower part is not casted perfectly. Many times it happens that when you run SDS Page, you will get protein like this. You will get protein like this. What may be the reason? It means the transition which you have made between stacking and resolving is not perfect. That's why it's accumulated at the top itself. Many times you will get result like this. What may be the reason? The reason is the time for the run was 2.5 hours, but it was overrun. So all protein they drained into the buffer. All protein drained into the. So these are some of the situations why I have told this, because when you run this experiment in your lab, these problem comes, and it's very difficult to troubleshoot, right? So many times, one last situation I'll give, because this happens commonly. Now you'll get protein like this. This happens commonly with all. Now you have to tell me what is the problem. Very light faint band, protein is there but it's very faint, light. And this happens commonly with all. If any one of you have done SDS Page, maybe you have encountered such type of problem.
Limited quantity of
No. That may be one of the reason, but that is not the reason in this case. No. That may be also one of the reason, over destaining. But in such cases, see, when destaining is more, that you can easily make out. But in such cases, when you stain it, first go itself, you will see that it's very faint band, and then resolution is not very high, it's very fuzzy band. The reason is, as I told when I started SDS Page, difference in the pH. So you will get fuzzy band. Rest other, that easily you can make out, like if there is over destaining, so easily you can make out that earlier it was very high staining, now it's not visible, so again you can stain it. Okay, so it is the pH, because of which resolution will be not high, so fuzzy band will be there. Last point, for staining it takes almost 3 hours, 2.5 to 3 hours, but if you heat your gel, that gel in a buffer or water for maybe 30 minutes, so within 30 minutes you can stain and destain. So you take the staining solution, for 2 pulse you put it in micro oven, for 2 pulse you just heat it and then keep it in destain, so easily you can see the appearance of the band. So this is to cut short the time, right? So this is in brief about the SDS Page, and maybe during experiment I'll brief you about the 2D Page, right? So I'll stop here. Thank you.